Okay, today we're going to talk about leak channels and about their effect on different parts of the eye receptors or the photoreceptors and the bipolar cells. So, let's say we have some photoreceptors or bipolar cells here. We'll put some channels here. We'll go chlorine here, potassium here, and sodium here. So, this is the ICF of these, and this is the ECF. We'll put ICF here as well, and ICF here as well. So as we know, chlorine tends to accumulate more on the outside of the cell, in the ECF, than on the ICF part of the cell. So if we were to create chlorine as a leak channel, that would allow it to go in and would cause the RMP to decrease, causing a hyperpolarization of the cell. And that happens because of the effect of chlorine in the more positive environment of the ICF.
Okay, question. What is the meaning they increase the conductance of the leak channel?
When you increase the conductance of a leak channel, that increases the ability of an ion to go in and out of the cell membrane, or
Okay.
through the cell membrane. So the permeability. So if we do that for something like potassium, that tends to be more inside the ICF of the cell. If we were to create a leak channel of it, it would actually rush out, unlike the prior example. This would also cause the RMP to lower, causing another hyperpolarization of the cell because the potassium is leaving and the gradient is going down of the energy. Now, unlike these, sodium also tends to be more on the outside in the ECF than the ICF. So if the leak channel were to happen, it would rush in, but unlike chlorine, it would actually cause the RMP to rise. And with the rising of the RMP, this depolarizes the cell.
Okay, then those three cells are photoreceptor cell, and then what would be the result of it?
Yeah, so if we were to take these as photoreceptor cells, what would happen would be that once the chlorine channel leaks in, it would cause a decrease in the amount of glutamate released from the photoreceptor cell to the bipolar cell, which would help us see light and would cause action potential to happen because it would stop mGluR6 from closing the TRPM channel. And as for potassium, it would be the same with the leak channel. Since the RMP is going down, that would also stop the glutamate from being released as much into the bipolar cell, which means mGluR6 can still not close the TRPM channel. And as that happens, sodium is actually the opposite. Because the RMP is being raised, it allows glutamate to be released even more into the bipolar cell. And so when that happens, that would cause us to see dark because the TRPM channel would be closed and less glutamate would be released to the ganglia cell. So you would see light with these two and dark with this one.
Okay, and those cells are bipolar cells, and then what will be the result?
Yeah, so if these would be bipolar cells instead of the photoreceptor cells, it would do the opposite. Since these are causing less glutamate to be released from the cell, same would happen for bipolar cells. Since less glutamate would be released because the RMP is lower to the bipolar cell, that means we would not receive enough in the ganglia cell and we would deem the light that we see as dark. And sodium, since the RMP is raised, we would release a lot of glutamate, which would allow the ganglia cell to receive light and have us think it is light.
Okay, please tell me the difference between metabolic channel and inotropic channel in bipolar cell.
Yeah, so there's two different types of channels that we can encounter there. The first one is called the inotropic channel. So the inotropic channel is a channel that causes an increase in activity. An example of that channel would be the AMPA channel. Since when the AMPA channel receives glutamate, it allows something like the TRPM channel to open. It causes an increase in the activity, while the difference with the metabotropic channel, this is a hard word, is an example of that would be the mGluR6. mGluR6, what happens when it receives glutamate is instead of keeping the TRPM channel open, it closes it. It causes a decrease in the activity within the cell. So mGluR6 decreases the activity, which decreases the glutamate that is released, and that causes us to see dark instead of light. And so these are the two main differences between these. Metabotropics, I guess you can say, would hyperpolarize the cell, while inotropics would depolarize the cell.
Okay. Please tell me the difference between rod and cone and how they are related with inotropy and metabolic channel.
Yes. Okay. Let's erase this part real quick to add more room. So as we know, the back part of our retina, to the place where the photoreceptors are, tend to have two different types of photoreceptors. The first one is known as a rod. So rods tend to be connected to metabotropic cells. They also are known for helping us see black and white. They're not able to see color and only can tell shades in a great cell. These also tend to slow things down, and they also
What slow things down?
the metabotropic channels in the rods.
Okay, so what kind of metabotropic channels are in the rod?
Rods, you have the mGluR6.
Okay, so angular 6 the one that slow down
Yeah.
the activity. Okay.
Here you go.
Mhm.
Yeah, and rods also are known as rhodopsin cells. And so then you also have cones that, unlike this like hard top of the shell or of the rod, tends to be more of a pointy top. And these cones help you see colors in the RGB scale. So that's red, blue, and green. And they're more known to work with inotropic cells, like the AMPA, or inotropic channels, like the AMPA channel.
Where can you find mostly rod cells? Where can you find most of rod cells? Where's the location of rod cells?
Yeah, so the rod cells, you tend to find them in
That's
the
cones.
whoops, wrong one. Yes, the rods. Rods you tend to find in the periphery, in the peripheral parts of the retina.
Okay.
While cones, instead, you find them in the center of the fovea area of the retina. Thank you.